Good day, everyone. I'll be presenting a part of my MSc project, which is testing the utility of DNA barcoding in Diptera of eThekwini. So Diptera, the order Diptera includes all true flies, such as your crane flies, your rubber flies, fruit flies, and mosquitoes, to mention a few. This order is extremely diverse and is one of the most taxon-rich animal groups in the world. The Diptera play an important role as decomposers and pollinators, so they help in the functioning of the ecosystem. So for biodiversity assessment, species identification is very important. Knowing what species exist in a particular habitat helps in conservational planning. And usually this is done using taxonomy. However, this method is time-consuming and it also requires well-trained taxonomists. So DNA barcoding provides a method that can complement taxonomy, and it uses the short CO1 fragment to identify species. So the success of DNA barcoding largely depends on the absence of the overlap between intraspecific and interspecific variation, a concept known as DNA barcoding gap. So for this barcoding gap to occur, the amount of genetic variation within species needs to be smaller than the amount of variation between species, and this allows unknown sequences to be assigned to an existing species or flagged as a new species. So DNA barcoding has been successfully used across a number of insects and has been also used to identify species belonging to fish, birds, amphibians, reptiles, and plants. Within the insect group, the efficiency of DNA barcoding in identifying Diptera species is still being questioned. A paper by Meir et al. Reported a low identification success rate because they had less than 70% identification success rate. However, their study didn't include any of the African taxa, so it is unknown how DNA barcoding will perform on our South African taxa, which brings me to the aim of my study, which was to test the utility of DNA barcoding in the Diptera of eThekwini, where we first had to look at the barcoding gap. So is there a barcoding gap within our South African taxa? And also look at the efficiency of the CO1 gene as a marker for identifying Diptera species. So we collected 1,060 Diptera specimens across several reserves within the eThekwini, and these specimens were photographed, and we chopped off a leg and we sent it off to the Canadian Center for DNA Barcoding, where it was sequenced. We had sequencing success. 844 specimens were successfully sequenced for CO1 gene, and they belong to 44 families and 214 genera. So for DNA barcoding gap, to look if we have the DNA barcoding gap, what we did, we aligned all 844 CO1 sequences using the Clustal W module in BioEdit, and then we calculated the interspecific and intraspecific variation using the K2P model in Spider, using the Spider software in R. And what we found, this is just the summary of the overall variations. We had the interspecific variation ranging from 0.0 to 0.23, and the intraspecific variations ranging from 0.0 to 0.28. And you can just see from here that the variations are much larger within species than between species, which means that there's actually no barcoding gap. And we plotted these interspecific and intraspecific variation. So we plotted the densities, and just to see the overlap between these intraspecific and interspecific variation, as you can see in that graph there is actually no barcoding gap. So the next step was to now test the efficiency of the CO1 gene in identifying species. So we used 3 distance-based methods, which are also found in the Spider software in R. The near neighbor, the best close match, which was used in the Meir et al. Paper, and the bold identification criteria, which is usually used in barcoding studies. For the results, we had different species assignments. We had different percentages. For the near neighbor, we had an identification success of 68%, and for the best close match, we had an identification success of 65%, and for the bold identification criteria, we had an identification success of 62%. This means that the sequences that were correctly assigned to their species names. So for our discussion, we see that there is actually no barcoding gap in our sequence divergences due to the high interspecific variations that occur. And our study is similar to the one that was published by Meir et al. In 2006, where they reported an extensive overlap in Diptera, where 99% of their pairwise distances for congeneric sequences fell into the area of the overlap. For the identification success, we had varying percentages for the different distance methods used. The near neighbor had the highest identification success at 68%, and the bold identification criteria had the lowest identification success at 62%, and these results are also similar to those obtained in the Meir paper, where they had an overall identification success less than 70%. So in conclusion, DNA barcoding is a great tool that can be used to complement taxonomy. However, due to the high intraspecific variation that exists in Diptera, it poses a problem because there is no barcoding gap. So this lack of barcoding gap and the low identification success suggests that the CO1 gene is not a good marker to use for Diptera, and any identification using the CO1 gene can lead to misidentification. Therefore, we will need to look for other genes that we can use to identify the Diptera species. I'd like to thank the following, and thank you.